Okay, welcome everybody. It's a pleasure today to have Andrey Bagrov here from Radboud University in Nijmegen. And he will tell us about his work on the multi-scale structural complexity of natural patterns. So
thank you, Wout, and thanks, Fernando, for inviting me here. I really appreciate this. And indeed, today I'm going to talk about maybe the main concept in the whole science of emergence about complexity. But what is complexity? In principle, it is something that we immediately recognize when we see it. For example, when we deal with a biological cell or some beautiful fractal or whatever. But it's very hard to define it formally. For example, Ted Lloyd, in his small note, collected about 40 different definitions of complexity. And of course, I think there's very little or no hope to come up with a single unique correct definition. But still, it makes sense to discuss it. And in principle, all of definitions can be crudely decomposed into two broad categories. Either it is about computational or descriptive complexity, or it is about more emergent stuff, effective or physical complexity. So descriptive complexity is easy to understand if we look back at the very first probably definition by Kolmogorov, that complexity of a structure is the length of the shorter description in given language, which you need to define this structure or object. For example, it can be a depth for quantum circuit that you need to generate your target quantum state starting with some simple reference state. Or it can be a length of an instruction which a computer program needs to decompress a file. And in principle, there's nothing wrong with this computational complexity. It's a respectable field in computer science and it serves many purposes. But when it comes to discussing kind of more emergent, more structured stuff, it is probably not the best way to approach it. Because, for example, from a point of view of such computational complexity, the more random thing, the more complex it is. And according to those definitions, random white noise would be a way more complex object than a beautiful painting. Or, for example, when we move to the realm of biology, then complexity of genome of a simple flower would be way higher than complexity of genome of a human being. And I'm not very happy with this approach. So when we ask about complexity in nature, we want to have some kind of definition of effective complexity, which maximizes itself between fully ordered structures like crystals and fully disordered and highly entropic like gases or random noise or whatever. And it is not just a philosophical question, from my opinion. Because, for example, if we want to come up with a more quantitative theory of evolution and want to characterize different stages in biological evolution, for example, stages of continuous development and major evolutionary transitions, like from prokaryotes to eukaryotes, we need to have some notion of complexity. Also, we want to understand how our brain works, and we as human beings like to discuss complexity. So what exactly our eyes and brain view as complex systems? Here we can bridge psychology and some formal theory of complexity. Also in neural networks, like modern approach to neural networks, to AI is very instrumental. Like we want to develop networks, algorithms that work better, that solve concrete problems. But as scientists, we also want to understand kind of what is this structural effective complexity of networks, why some networks work better than others. Or, for example, if we want to develop hardware neuromorphic matter, then maybe we can use some principles of complexity as a guidance to developing such matter. And our main idea is to try to relate complexity to self-dissimilarity of an object. Because if we look into nature, then often when we deal with a complex system, its complexity means that it has many levels of organization, and also that those levels are organized in very different ways, like levels of organelles or cells or tissues or populations. They're very different, and they're not similar to each other. And this approach is somewhat complementary or opposite to the concept of self-organized criticality, which I'm sure many of you are familiar with, introduced by Per Bak and his collaborators. And he wrote this very ambitious book, How Nature Works, when he tried to relate complexity to scale invariance, to criticality. But apparently, it's not quite the case. Because in nature, we have self-dissimilarity instead of self-similarity. Of course, this research program is very ambitious, and we need to make a first step. And the simplest object we can start with is just 2D static patterns. Just patterns we see in nature, images, photos, whatever. And the idea is the following. Assume we have a pattern, and let us define some coarse-grained procedure on this pattern. It can be as simple as just window averaging, so that you start with a picture like 2024 by 2024 pixels, then you average it by windows, like windows 2 by 2, and then you get another image with 512 by 512 resolution. And then you see how much information you lost during this renderization step. And then you do it again and again and again, and then you compute overlaps between subsequent steps of your RG flow. And if the overlap is close to 1, then it means that you didn't really lose anything. And there is no complexity contributing at this scale. But yes.
And how do you quantify this loss of information across scales?
Yeah, okay. There are many ways to do it, but here we just literally compute overlaps of 2 pictures. It's like you have intensity function. So assume it's just grayscale pattern. So each pixel has its intensity. So it's a function at step k, function at step k plus 1. So we can compute either L1 norm or overlap or stuff like that.
But they are different dimensions.
You mean that when you go from 2000 to 500?
Yes.
Or, okay, then you can just upscale it back. So.
Oh, okay.
But maybe you can actually be more specific. Do you actually have some sort of a residual entropy or something?
Yeah, you could, yeah.
When you go from between scales,
Because
so there's some something like that.
they could make the divergence between each scale.
Yes, it's also possible. So indeed there is so this kind of definition is an umbrella-like definition, because we can use different approaches to coarse graining, and we can use different approaches to computing kind of dissimilarities. And I fully agree that this entropic approach is also can be very fruitful, but just in our simulations we did simple deviations from perfect overlap. So there are some ambiguities, but we can stick.
Just to be clear, what you're doing in this case with the scaling, then you just compare the variance within the 2 by 2 cells with its mean.
Basically, yes. And then we go step by step. Okay, so if we stick to some concrete procedure within this umbrella definition, we can try to apply it to different systems. And as physicists, I come from condensed matter physics background, so we like to work with systems like Ising models, magnets, Hubbard models, many-body quantum stuff. So let's start with something very simple, like the Ising model, and consider the phase transition between ferromagnet and paramagnet through a critical point. And then, indeed, we see that complexity of those images undergoes certain pattern, with ferromagnet being very simple, critical point being, in this case, the most complicated, and then slow fall off into the paramagnetic phase.
What you mean? Just to compare to the image, you create a photo with two colors for the face.
Yes, so we do in this case some classical Monte Carlo simulations at a certain temperature. We pick a particular snapshot and a typical representative at this temperature, and then it's Ising model, so it's black and white or red and blue. So we assign numbers like 0 and 1, and then we compute complexity.
But you do it for different scales, so you make it larger and larger and larger, and then you
Basically, we do it. We generate the snapshot for a rather large system. I think in this case it's about 4096 by 4096, and then we do this coarse graining and
Fixing the temperature, so you.
yeah, we fix the temperature and compute complexity. And
yeah, it's not so clear. So what is the definition of the complexity? Is it this formula on the?
yes.
This.
Okay, so maybe I can
Scale?
elaborate on the whiteboard on the our online attendees will
Can
be
use the whiteboard in the.
maybe if you can help me with this. Oh,
Do you wanna write on this? Yeah, but they won't even see. You will see it if you're writing on here. Hang on, please.
okay, just on top of the slides, right?
Yeah, yeah. I think this way, and then you can write on the black picture.
Okay, good. So your question was like
Now it was about the definition. So you have this plot here, which clearly
Yes.
has a peak.
yes.
So what is then?
uh,
How do you define
Sure.
C as a function of?
here I have two plots. This red curve and blue curve. So in both cases, I take my image at a given temperature, given snapshot of my Ising model, and then do this coarse graining with averaging intensities in 2x2 windows. Then, so for example, I do one step. Per segment, and it had white, black, white, so forth. Then I average it. So, and here within the 2x2 windows, I had, for example, 1, 1, 1, 0, and now I have just 0.75 intensity. It's the most naive procedure you can imagine. And then I compute 1 minus overlap between those images.
And then you assume that the highest scale or.
And then I sum over different case. So in principle, so this particular overlap or deviation of this overlap from 1 would be C naught in this notation.
Okay.
Then I do it again.
Yeah.
Again, some C1 and so forth.
But you have to do some normalization, no? Because
Yes,
otherwise-
I do some normalization at the very beginning. Okay, and then the point is
So
that
sorry, in the plot here, the blue is the complexity measure, and the red is just the
no.
magnetization.
Look, so both red and blue are complexity measures, but in one case I sum from the very first scale, and in the other case I ignore this microscopy because I'm saying that okay, maybe just very short range patterns are not relevant. Yes, when we speak about complexity, it's not only about having many scales, but also about thinking which scales are relevant for us and which are not. And we know that if we have some random noise, some power magnet, we'll have a lot of patterns at this pixel scale, but if we just average it a bit, we get some homogeneous background. So indeed, if we incorporate this very first scale, then complexity just maximizes. But if we ignore this very short scale contributions and see what's going on, then we see that complexity would maximize around the critical point where patterns are actually more complex.
So you say the first scale does not allow you for divergence, but just
Yes, so it's a kind of heuristic rule, but of course you have some freedom here, which scales you're interested in and which scales you're not interested in. But in many physics applications, as we discovered, it's always better to ignore, like if you're interested in some maybe unconventional orders, is to ignore patterns which are specific for the very microscopic scale, because they are too random.
So what happens if you start at the second scale? If you do case
Okay, then it would go a bit lower, but then this will be more pronounced.
Okay.
Okay, and we see here that complexity is really sensitive to the critical point, and we can see the transition line, transition point, without computing any correlation functions, without knowing the order parameter, just from structural complexity.
just quantifying the overlaps across each scale.
Yeah, across different scales.
So any function that connects or quantify the similarity.
The similarity between.
But this is because there is a diverging length scales correlation.
Yeah, but there will be more non-trivial examples, because we started with the Ising model because it's simple,
I know, sure.
but now we can consider something more sophisticated. And the next example is how do I erase here? Thank you. Okay, now again, let us consider a classical magnet, but now it is a Heisenberg model. So degrees of freedom are not just binary, but they have some continuous rotations. And we have Heisenberg exchange and DMI interactions in external magnetic field. And then we can get more complex patterns in such systems. So for example, we can have so-called spin spirals or magnetic labyrinths. We can have Skirmani crystals. So here the plot is small, but it actually has been simulated for large lattice, and those are really skirmions. We can have some transition, which is called skirmion bimeron mixed phase. And here, if we now complete this complexity, where again green and red line are complexities incorporating all scales but the smallest one, then from the derivative of this complexity, you can see transitions from magnetic labyrinth to skirmion and from skirmion to ferromagnet. And while this one is very trivial because it can be captured, for example, with monetization, this one is not trivial at all. And if you do not know which order parameter corresponds to it, then from simple observables like monetizations or standard two-point functions, you will be unable to deduce it. So we thought that, okay, maybe complexity is a very good tool to sketch phase diagrams of complex systems for which we do not know order parameters and symmetry breaking patterns.
But here you use the derivative of this function.
Yes.
In the previous one, in the Ising model, you just use the function itself, as a measure, as a definition of complexity.
No. The definition of complexity is still this one. The question is, when you have this complexity curve, you can also do some manipulations on it to deduce.
Okay.
Like.
But I mean, following up on that question, in the previous slide, you had this.
Yeah.
If you take the derivative, you get a pole with negative function. It looks completely different if you take the derivative.
Yes, because those correspond to different types of phase transitions. When you have this ferromagnet to paramagnet transition in the Ising model, it's a very conventional critical point, like textbook phase transition. When you go, for example, from magnetic labyrinths through this intermediate phase to carbonic crystal, then there's no critical point. This correlation scale does not diverge at the critical point, at the transition point. And it is more like kind of structural transition. Typical lengths here, here, and here are kind of the same. The radius of fermion is comparable to the diameter of the Vimeron tubes, or
So you're saying you can, from these curves, you can also deduce the type of phase transition?
in principle, yes.
And if I understood correctly, somehow we're integrating all these scales and quantifying as a number. And do you have weights for each scales, or
Here, we do not assign different weights to different scales, but you are right. What is more interesting is not this overall number C, which is a sum over CK, but rather the distribution of partial complexities over different scales.
Yeah.
And that's exactly what I was going to show on the next slide. That if you plot this CK in different phases with a function of K, with a function of scale, then you can see, for example, that at the critical temperature of Ising model, okay, you have some microscopy, but then you have a nearly flat scale invariant distribution of CKs, of partial complexities. In paramagnet, complexity dies off when you go to larger scales. And for spin spirals or skyrmills, there are some typical characteristic lengths which characterize those patterns.
well, you're saying if you check in isolation each length, that's like the most important landscape?
Yes. And as I mentioned, this transition from spin spirals to Skyrmilk crystals is not that easy to detect with correlation functions. So, for example, here in different phases, like this one is more, A and B are actually the same, but with a bit different symmetry. C is bimeral phase, and D is, let's say, skyrmills at high temperature when they become quite noisy. That charge factors here and here are very similar. So if you just look in the Fourier space, you would say that, okay, there is kind of some difference. This is circle, but here we have kind of the same symmetry, but a bit noisy. But those are two very different phases. And in this complexity curve, we see it. And just in Fourier space of two-point function, we do not.
Which patterns on the left, which curves on the left correspond to the two circular Fourier spectra?
The red.
Yeah. So this one is red. This one is orange.
The ones that
The red and orange are the same.
They're similar, but okay, you need kind of here to combine this interscale distribution, this CK profile, with the dependence of overall complexity on your physical parameters. And then you can really see transition points.
But I don't quite understand. You might make the point that if you look at the correlation function, you cannot distinguish between the left and the right patterns. But that if you use this partial complexity, you can. But these two curves look pretty similar. Quantitatively, they differ a little bit, but
It would make more sense if, for example, if you would compare the blue with the red for left versus right. The same.
Okay, so you mean that if I had correlation functions for blue and for red on the...
I think what we're trying to go to, you have this pattern A and you have this pattern D.
Yes,
and they have a similar Fourier spectrum.
yes.
So you say you can't use Fourier spectrum to distinguish these two phases.
Yeah.
And then you say, but we can't use complexity to distinguish these two phases.
Yeah, but the point is, okay, I do not show it here, but like if we get, sorry, one slide back. Then like the dependence of complexity on magnetic field is rather smooth, so we have some kind of dip here. But then once you're in a given place, or for example, you have Stirling crystal at this point, or
So you mean the absolute value of the
yeah,
complexity?
absolute value,
Right. Then when
it doesn't differ much, but when you compute complexity for different snapshots, it's very stable. So it's stable like within some digit after the comma. And as a result, so like each curve here has been computed not for single snapshot, but for many snapshots. And error bars are within those marks. So it means that if you do your computation here and do your computation here, the numbers will be in principle quite close, like 0.5, 0.4. But within error bars, they are very distinguishable.
Okay. So if you go to the next slide, you see these two curves. Are these normalized curves or non-normalized curves? Because if you add them up, they will add up to the absolute value of the complexity. Because
yes,
that would be the idea then. So the
yes,
orange one is lower than the.
yeah, and
That's right.
so we do not normalize complexity, we normalize intensity of the original pattern, and then we go through this coarse gradient flow.
Saying it is not just complexity in this left plot is not just about the shape, it's also really about
Magnitude.
the magnitude that
Yes.
you see. Ah, okay. So
It's
you can
about the
distinguish, so
the-
the difference between 1.5 and 2 point,
Mhm.
0.15 and 0.22
Mhm.
is really a
Yeah.
significant difference.
Yes, of course, like the absolute value of complexity per se doesn't mean anything because it depends on the procedure. But once you fixed the procedure and you stick to it, then relative values of complexity, they matter. And even if you have a small difference, it tells you quite something because complexity is very stable. That's what we see from our numerical simulations.
And one question for the paramagnetic in the schema, because you don't have log plots, but these are also scaling variant or not?
Sorry?
The paramagnetic in the schema crystal, they seem to decay like but I don't know exactly how.
You mean that it looks like power law?
Yeah.
Now here it is a different story because if you have power law in the correlation functions, like in a critical point,
Yeah.
then most likely complexity will be very similar at different scales. So your familiar power law, the critical point would mean kind of nearly constant behavior here in complexity.
I see.
Then, because for the two cases here, for the first case, the Isaac model is important to ignore the first K and only look at later values of K. And for the other one, the beginning is more important. So if there's some.
First of all, nowhere here the first point is important. Like in the Isaac model, it contributes a lot, but it's more kind of a psychological reason to just throw it away, because when we see this paramagnetic pattern and it's quite random, we want to exclude randomness from our structural complexity. But in principle, you may exclude or not exclude it. You just need to be consistent in what you're doing, and then you'll still be able to see this transition points or signs of criticality.
Yeah, so I mean, just a final clarification then.
Huh?
So what you said in the beginning was that if you include every scale, the Ising model is constant after the critical point, above the critical point.
Yes.
Constant. And only if you ignore the first level, you
Yes.
get this decrease and this maximum. So this is actually akin to, or exactly the same as removing the first point.
Yes.
And then all these curves actually add up to the same value. That's basically the final. No?
No, they don't. That's the thing with
But the green and the blue will add up to the same value.
yes.
Right.
Like the green and blue in the Ising model, yes.
And how do you, of course, in simulations you're finite, but how do you use this thermodynamic limit? Because like in the harmonization group, you do it infinite decimations until you stabilize, etc.
Mhm.
When a real system has 6, 10 to 23 instances, like atoms,
Mhm.
or the magnitude, so then ignoring one variable in a thermodynamic limit is not natural, because it's like it's so big anyways. So how do you adapt or make your choice consistent with that?
Sorry, I'm not sure I understand your.
So I'm saying if you go to the thermodynamic limit,
Yes.
ignoring one layer is not, there's no meaning because it's infinite anyways. So then like if you think about atoms as 6 to 10 to 23 variables.
No. It really depends on the phase in which you're considering your system. Because if you're constructing RG for a critical point, then of course, like you have like infinite number of scales and there's scale invariance. But if you, for example, in a system where you have very short range order, then one scale can be important.
I have a more philosophical question
Yes.
about, because you started out by mentioning a biological system from molecules to atoms to cells and networks. The pictures you show there can be generated with a model for vegetation dynamics under different rainfalls.
Yes.
The system doesn't change except the rainfall. So are we talking about the complexity of the realization of the system, or are we talking about the complexity of the system?
Okay, here, since I started speaking about static patterns at this part of my talk, then of course it's complexity of a particular realization, because when we look at the static picture, we cannot really judge what the system is capable of in other regimes. But in principle, and later in the talk, I'll also speak about more general systems. And there, if you incorporate kind of general dynamics into your structure and define cross-graining on the whole system, then but of course many things are still to be done.
Yeah, I'm sure. But it was more like, you know, here we look at the realization.
Yes, here we speak about
the system that are we talking? Is that what we really want to think about when we're talking about complexity, or are we talking about the complexity of the system?
Okay, but in principle, I think complexity of a system can be also related to complexity of its realizations. It's like here, if I scan over like the whole phase diagram of some system and do not see a single complex phase, then the system is simple.
That's true.
Yeah, okay, so no. Also speaking of this complexity maximization for not most entropic configurations, we conduct a very simple experiment. So we just put a drop of food dye in water and let it dissolve and make photos for this process. And indeed, we know that, so here, okay, the drop is even invisible in the very beginning, then it starts dissolving, forming some complex, beautiful patterns, and then eventually it reaches this homogeneous distribution. And we know that entropy of this process, we cannot measure it, but we know that it goes from very low values to the maximum. And here in dynamics, we consider that first it maximizes around these patterns, and then it starts dying off as the system becomes more homogeneous and kind of less interesting to our eye.
Just as a
Mhm.
curiosity, you say entropy always grows, but have you looked at entropy production or entropy rates?
Oh,
Because it's an irreversible process, so you have entropy production, which is the governing effect.
it's a good point. Well, we did not.
Might be interesting to.
Mhm. Yeah. Okay, and then so far I was speaking about just
So for that drop, which order of complexity was the most relevant? Oh,
you mean in this CK distribution? Like,
Yes, yeah.
oh, I'm afraid I do not remember. Like we didn't put it in the paper and I forgot it. But I agree it would be interesting to see how this distribution between scales evolves in time.
Yeah, yeah.
Like,
Did you again subtract the lowest order here or not the lowest step?
Uh,
Or ignore the lowest step here?
Hmm?
Did you again ignore the first step here?
honestly, I do not remember because this has been done by our collaborators, so we put it in the paper, but
Because I was just wondering if you would include basically, yeah, you didn't remember, but if you would include the first step as well, that maybe also again
Mhm.
levels up, or is that not possible?
it should be possible, but I think we need to just gather this data and compute it again. It's easy.
Yeah. But here it wouldn't matter, not because it's depending on the resolution of your photo, your picture. Yeah.
Yes.
If you take a better camera, you should get the same result. I mean, that's my guess.
Yes. So here we have some natural cutoff which is given by resolution. So indeed, it's like scales we cannot access, we consider as irrelevant.
Right, exactly.
Yeah, of course, in the last picture, if it's completely homogeneous,
Mhm.
then of course your lowest step indeed
Mhm.
doesn't give any information, so then yeah, it shouldn't matter, no.
Okay. And then we apply this complexity to some experimental data. So it was a very simple question asked by our colleagues from scanning microscopy department, because they studied phenomenon of emergence of magnetic order out of spin glass when you raise temperature in elemental neodymium. So it's a bit counterintuitive phenomenon then that normally we think that when we cool our system down, some phase transitions into more ordered phases occur, but the surface of elemental neodymium and opposite thing happens. So that of course at very high temperatures you have disorder. When you cool it down, you get into an ordered phase. So here it's maybe not very visible, but like those large patches here, they have different orders, but within the patch, this order is kind of similar to antiferromagnetic. It's very simple. But then when you cool it down even more, you enter a very random spin glass phase. So they found an explanation to this phenomenon, but what they were also interested in is can we detect this transition point in an automated way without kind of looking at these snapshots of STM, without training neural networks to distinguish them. And the answer was yes. So again, we computed this complexity and some alternative version of it. I'll explain it in a second. And in both cases, we found in an automated way this transition point around 8 Kelvin. So just by feeding STM pictures into our definition of complexity. And here we have some consistency. So on the left plot, we have this interscale dissimilarity, which I've been talking so far about. And on the right, we use some kind of alternative definition of complexity when we kind of focused on a fixed scale and computed similarities across image within the same scale. So we have some high definition, high resolution image. Then we took different patches of the same size from this image and computed the similarities between those patches without doing any cross grading. And then obviously, again, if you have very simple order, all these patches will be identical. If you have disorder, then probably they will be kind of more or less on the same distances from each other. And if you have some distribution of complex patterns, like one pattern here, another pattern here, third type of pattern here, then you get some non-trivial distribution of dissimilarities. I don't have an equation here, but we kind of came up with a single number for the distribution of the similarities between different substructures within our picture. And again, we got some clear transition point. So
Could you explain the difference between the two?
Okay, so this one is what I talked about before. So we do cross grading, we compute the similarity between different scales. Here, we just take our image in its original resolution without cross grading. Then we sample sub patches from this image and compute the similarities in the same way, like overlaps between.
But what is the choice of your sample? In this letter, you have to sample a certain way.
Yes.
Does that matter for the picture that you get?
It affects, of course, this absolute numbers.
Right.
But in principle, it doesn't matter. We didn't play a lot around with this, so we just considered like 3 different page sizes, like 64, 128, and led to very similar results.
And when you say phase transition is visible, you knew a priori this transition point, and then you make this as fit, or you put direct from the data that is?
No, directly from data. So we kind of knew that there is transition by looking with our eyes, these pictures. But
did you also compare to the standard, say, Fourier space analysis?
Yeah. Basically, it is. Yeah, it's shown here. So indeed, in the Fourier space, when you have large patches of a well-defined order, you have less singularities in the structure factor. In Q space, here we have more. But also it was kind of nice. Okay, you can use either approach, complexity or Fourier, but it was just nice to see that it also works.
No, I'm just wondering if you should do.
And could you also do the whole thing in the Fourier space, like trace the evolution on the Fourier space?
Yes, and in fact, we are doing in different contexts. So I'll get to this point. Okay, so this part of the story was about classical systems. What about quantum ones? So for quantum system, it's kind of harder to come up with this cross grading defined for a quantum state. Now, of course, you can have RG flow in quantum field theory, but for example, if you have a finite size many body system, for example, the one you simulate on a quantum computer, then cross grading for a wave function with a fixed number of sides is a not very good defined procedure. So what we decided is to resort to our classical prescription. So we considered a particular problem of certification of quantum states which you generate on a quantum computer, because we know that quantum computers are not some magical machines. There are errors, there is noise, and even if you just wanted to generate a particular target state, you need to be sure that it generated something close to its target state and not some crap. And that's the whole task of certification of quantum states. And when you do the quantum computer, so you have some reference state, then you have quantum circuit which generates your target state, and then you can do local measurements on your qubits. And from these measurements, you get some snapshots, which basically look like bit string arrays. So you did measurements of number of qubits. So in this case, it's 8 qubits, and you get some output, like spins up, down, down, up. Then you repeat your measurement, you do the next shot, and again, you get some bit string. And the question was, you have access to these bit strings, and basically that's the only thing we have in real quantum simulations. Can we certify the state that we get out of this simulation? And here we did, again, a very simple, very naive thing. So we assume that, okay, we can do a bunch of shots, a bunch of measurements in our system. So we can then concatenate individual bit strings and get just a long one-dimensional array. And then we can compute the dissimilarities within this array. And of course, when you, like, if you do it in a simplest way with averaging with window 2, then for first few steps, you compute the similarities within individual shots. Then you go to a larger scale and you compute the similarities between randomly concatenated shots as a whole.
Another question, because that way it seems that the similarities only connecting neighbor scales, but you could have a full network of similarity from the higher to the lowest scale. Is it interesting, or you lose a lot because it's too many scales?
You mean that if I do not sum up and
No, I mean, like, you can get the similarity between the last scale and the first scale as well, but
yes.
how much do you lose? Or is this interesting because you can create a network of similarities?
No, I agree. It is interesting. So in some limiting cases, it will trivialize. It depends on your RG procedure, the coarse graining. But yes, we can consider it. Just so far, it didn't seem to be important. Okay, and then we can do measurements in different bases, right? So at least we have this freedom of do on-site rotations in our quantum simulator, and we consider two options, so that either we measure in the Z basis, base of local eigenstates of sigma Z operator, or in a random basis, where you randomly rotate each basis of each side of each local qubit. And then what you get in this way. So here dk means the same thing as ck before. We just decided not to use word complexity in this paper because complexity in the domain of quantum computers means totally different thing, like circuit complexity. So we decided not to interfere, but we call it just dissimilarity. And for example, here I have plot for a particular class of states, so-called decay states, which are defined as following permutations in the space of ups and downs. And then you get a set of decays in one basis. Okay, this one is now hidden.
You can press the X, red X. Yes.
Okay, so those are the similarities in that basis. Those are the similarities in random basis. And then your state will be characterized just by this sequence of decays, which you can view as a kind of hash number of your quantum state. So that's something very easy to measure because it's only about local measurements, and you do not need to do a lot of measurements, a lot of shots to converge to some particular numbers. And then you see that, so this index D capital is the index of decay state. The different decay states would have similar but different patterns, distributions of this decay. And we explored different classes of states, and in this particular case, when we use two bases, Z basis and random basis, all the different classes of states fall nicely on a 2D plane. For example, here we have decay states, here we have states of quantum Ising model in a transverse field. Here we have Schrödinger cat states with JZ state, all ups plus all downs as the ultimate limit and a trivial one. Here are states in the cluster equivalent model, ground states for different parameters. So, and if you use, for example, some third basis, for example, sigma x, you add more to this diagram. And then if you do just a small number of simple local measurements and compute these dissimilarities, then you can do at least a partial certification of your quantum state. So your machine generated some state, you computed its hash number in terms of dissimilarities, and then, of course, if you find yourself exactly where you want to be, so for example, decay state with some index, and you get here, then it's not guaranteed that the state is precisely what you want to get. But there is a very high plausibility that it is. But on the other hand, as soon as you deviate from it, then you know that something went wrong. And in this way, you can certify states without doing kind of numerically heavy procedures like quantum tomography, full reconstruction of the state, and so forth. And what is interesting here, that doing this structural complexity thing, we can detect phase transitions of a more kind of complex and hidden nature than we saw before. So, for example, we considered the following XXZ Heisenberg model, spin mod half, which has two terms. Heisenberg exchange on odd links and on even links. So it's 1D, but couplings are alternating. It's red and blue. And it is known that in the plane of J prime to G and another P delta, this quantum model has several phases. And in particular, this, if I remember correctly, this phase one is a topological phase. And by doing measurements in two different bases, so in one basis, we compute the similarities and we can nicely track this phase boundary. Then we go to random basis and we can nicely track this boundary. So in this case, we detect topological transition in a quantum model, again, without knowing anything about what kind of topological protection it is, what is the order parameter of non-topological phase, and things like that. And this quantum story has been picked up by other authors. So now I'm going to speak about I'm not familiar with this auxiliary Zoom. Wait. Yeah, I want to get this left panel back to erase my.
No. Should do it again. Okay. Yeah, so no, that's twice. Sorry. There's
Now
no
a little green
the
arrow in the
left bottom
not
green
corner.
one.
Yeah, green.
where? Yeah, that's fine.
Ah, yeah. Yeah.
Thank you. It didn't work.
No, because this annotation is not in Zoom. These ones are.
Ah.
We have to deal with that.
We cannot.
Wait.
But you don't see them online.
So they don't see why? Okay, then it's fine. And I think we can.
The eraser, the thing above the green box, that's an eraser, isn't it?
Yeah, but it's not, that's for Zoom.
That's the Zoom erase.
Yeah.
You take one out, first scale out.
Okay. So what people did, they considered the famous Fermi-Hubbard model. So we have interacting electrons on a square lattice. So this first term is a Hawking parameter, kinetic term. Then we have repulsion between electrons, which is a kind of simplistic model for Coulomb repulsion. And then we have some chemical potential, which controls the density of particles in the system. And they considered numerical simulations. I think they did some quantum Monte Carlo, although I'm not sure how they managed with high density of holes, but also they consider some real-life quantum simulators for the Fermi-Hubbard model. And then they can obtain such two-dimensional snapshots of different local variables. So like Z-quantization, density of particles, spin polarity density of particles. And the question was, okay, we look at this kind of random small squares. What can we tell about the system? Can we see the transitions that we know that we have there? Can we compute maybe some important physical features? And since it's not a project where I participate myself, I cannot be responsible for every detail, and I can tell you everything. But as far as I know, what they did a number of snapshots, like we did for one-dimensional arrangements of qubits, and composed a large 2D patched picture out of it, and then computed complexity. And they did many things, but one of the most interesting things is that they compute behavior of complexity normalized in a certain way against behavior of entanglement entropy of a site in this model. And so entanglement entropy is in the inset and complexity here as a function of delta, which is 1 minus n. So basically that's the density of holes in the model. And you can see that up to normalization factor, the dynamics, the dependence of complexity on the physical parameter goes along with behavior of entropy quite nicely. And I don't have this plot here, but in fact, we also observed for some model states like Dickey states and cat states. So apparently, for a number of systems, it was shown that, of course, our complexity is not equal to entanglement entropy. Those are very different things, but it can be used as a proxy to estimate entanglement entropy behavior in a quantum system. Yeah, and also they computed it for different types of snapshots like Z-quantization, density, polarized density, and okay, if I plot something, it will stay with us forever, right? This one with zoom. So and here we can see. So, okay, so, I'm sorry. One more comment. So
So now you can go on.
So first they defined the typical scale. So basically that's where our complexity maximizes. Like before, Echelon uses CK plots, and it's very natural. Like what they did, they defined this average K, average scale index. And what they plot is 2 to the power average K versus density of holes. And we see that we have a very clear special point here, and special point here, which can persist for different temperatures. And quite surprisingly, this point is exactly, okay, exactly to the extent that we know the knowledge of the Howard model, where strange metal phase, this famous non-firm liquid behavior emerges. So that the transition from pseudo-gap to non-firm liquid. So again, we see quite important critical point just in the behavior of some complexity-related observable. Let's see. And with that, I like to change the gear and consider a totally different subject. That's our psychological perception of complexity. Because
Something
yes,
about the so you mentioned way in the beginning, right? So your measure of complexity, you would like it to be small if you're unordered, small if you're completely random,
yes,
and peaking somewhere in between where you make the transition from ordered to complex. So if I think about entanglement entropy, that also satisfies those criteria, right? So if you are completely random, there's no correlations between.
I'm not sure I would agree with you because, okay, at least in my opinion, entanglement entropy is in fact quite different from entropy in classical statistical physics. Because first of all, it characterizes not the system, because in classical physics, entropy characterizes the state of the system. Entanglement entropy characterizes also kind of the way in which we divide our system. Like
it's the entanglement between
between parts,
subregions,
between different subregions. Right. Okay, but
if the subregion, if the whole system is completely random, then
okay, but then
there's no entanglement between.
I would say entanglement can maximize because, for example, if you consider many body quantum states which are typical in the definition of Don Page, or have this saturated volume law entanglement, they are quite random. In the sense that probably there are not there's no well-defined order, no long-range correlations, but you have just volume law, huge entanglement, and it will be very very random, very disordered, and very entangled.
Did you check your measure of complexity for those type of
no, not
volume law, random anybody states?
No, but it definitely makes sense to do it. And especially, again, take a look at this distribution across scales, because probably we'll have some huge numbers around C0, around microscopic scale, and fall off in the rest. But yeah, I agree that it would be interesting to look. Okay, so can I proceed to the next story? By the way, how much time do I have?
Minus 2 minutes. But we can continue.
A couple minutes.
For a couple of minutes. Because I think I'm around the half of my topic. It's like I didn't expect such an intense discussion. Like, of course, like all the questions are very welcome, but yeah. Okay, so then just let me know when I need to stop, and I'll stop it in any moment. So
I like this part a lot. I'm a psychologist, but I had to leave in about 50 minutes.
Talk fast.
No question.
Okay, so So far, I've been speaking about physics, but one of our main motivations was just to understand it. Okay, as human beings, when we look at something and we say, okay, it's quite complex. No, it's random and non-structural. So what is psychological perception of complexity? And for that, we decided to engage in collaboration with real psychologists from Canada. And we picked up a particular data set, which contains about 1,500 images, which are categorized in 7 groups. So those are some natural scenes like that, suprematism, infographics, interior design, general art, which also intersects with suprematism category, advertisements, and some objects, which is a very broad kind of random category containing almost everything. And all those images were ranked from 0 to 100 by humans. So what they perceive as more complex or less complex. But this ranking was conducted in an implicit way. So humans were not asked to give a score explicitly, like this one is 87, this one is 32. But instead, they were shown a lot of pairs of pictures within those individual sets. And they were asked, like, what do you think is more complex? So they obtained a kind of network of orderings. And out of this network, they used some algorithm to assign kind of final scores to these images. So we have just this data set. We can compute our complexity for each figure, and we can do linear regression versus this human rank complexity. In this particular case, we did not use this averaging with small windows 2 by 2, but instead we went to Fourier space. Like we played a lot around with different coarse grain procedure for this project, and it turned out that if you want to achieve higher correlations with human perception of complexity, and then Fourier approach is kind of the most suitable one. So that we first go to Fourier space, then we cut out, we ignore higher momenta, and then we do Fourier transform back. And then we compare our figures. Okay, compare in like as before by overlapping them. Yes.
How were the humans chosen?
That I do not know, because this data set is something existing. So we're planning to conduct our own experiments with humans, but probably, like, if you're interested in, you can go to this GitHub link, and there is a description of the data set. Probably it is explained there. Okay, now I'll show you some nice correlations and some bad correlations and comment on them. So here I show 3 plots for suprematism, for natural scenes, and for advertisements. And you see that for suprematism, modulo some clear outliers, we have a rather good linear correlation with regression coefficient 0.82, with 1 meaning just perfect correlation. For
So
others,
correlation between what? I mean,
so we have our computed multi-scale structural complexity on the x-axis,
For the image.
and again, and human ranking. Okay, so I would say that we were quite happy with this figure. Outliers are outliers, they can happen, and we were not that happy with some others. And then I'm going to just verbally tell you about some very recent results that I didn't have time to put on the slides, because we improved over this. And the first point, why we believe we have way better correlation in suprematism than, for example, in scenes, is because here we have a very well-defined set of images. They are restricted by a very narrow class. And of course, if you see 2 pieces of abstract art, it's easier for you to compare the complexity than if you see, for example, just some landscape with mountains and a truck standing in the woods. And because of this implicit human ranking, that people were not asked to just give a score, but instead the score has been derived from these mutual comparisons, we believe that a lot of error here comes just from implicit variance in human ranking, which is not given in this data set.
And also each human was not the same person who ranked, it was a
It was a group of people.
So, but it would imply that if you would have some sort of measure of the variability between humans, they would say that in the left picture that the variability between different humans is smaller than maybe in the middle picture where different humans classify is different. There's a lot of variation between this, probably.
You mean that different humans were kind of involved in the ranking?
Yeah, so probably they asked a lot of people to look at this. And I guess that if you just take two suprematist paintings, that probably many people gave the same ordering.
Yes.
Well, in the middle case with the scenes, probably many people gave different orderings for the.
Yes.
I guess that's something that's underlying this or not.
But I think, okay, maybe I wasn't very clear, but that's what I'm trying to say, that when people are asked to rank very well-defined pictures, which are kind of comparable in some conceptual sense, then we get way lower variance than the category is very broad. But also, here we have kind of evident correlation, but not very strong one. But then we decided to study again distribution of complexity across different scales and see how different partial complexities, different kind of particular scales correlate with human perception. And again, it turned out that if instead of computing complexity over all scales, we deliberately ignore the scales which correlate poorly with human perception, then we can easily go from this 0.54 to something like 0.7.
You decompose your complexity in the most meaningful scale, and that one is the one you try to correlate with the ranking.
Okay, here it's the overall complexity just summed over all scales with the same weights. But then we looked at correlation at different scales, and those correlations were very different. So with like middle scales correlate quite nicely with like 0.75 coefficient, and some microscopic noise or very large blocky scales pulling this correlation down. So which also was found quite interesting by our psychology collaborators, that in this way we can say something about indeed what humans perceive as complex or not, on which scales we focus when we do this intuitive assessment. Okay, another project which I'm not involved in. Yes.
So when you do the analytical complexity,
yes,
if there is a Gaussian noise or any kind of other noise in the picture, that will be picked up by the analytical part, right? But might not be picked up by the human counterpart.
Yeah, but that's the point, that if you have some small noise, it will be picked up by the oral complexity, but also it will be picked up by this partial complexity at the microscopic scale, which I call C0 in the beginning of my talk. And that's the reason why we normally, we tend to ignore it, because we want to get rid of the noise. And then if you ignore the utmost microscopic scale, then you really focus on the structures.
Not least on the idea of the correlation, there is a recent work, where you collect neural spikes on mouses and only show images, and the authors correlate the perception space and the image with the spiking space. And it's pretty data, close question to what you say, but correlating the spiking directly with the image that is shown to the mouse.
So it was mice looking at images.
Yeah, and then you mice with probes, so they correlate the spikes of the mice with what they see.
Okay, because we'll have an ongoing project, but this one is on sleeping mice when we have just patterns of neural activations from sleeping mice, and we see that there are different stages in terms of intensity of activations and also in terms of complexity. So like most of the time nothing happens, then we have some complex but not intense pattern of activation, and then very simple but very intense pattern, and we're trying to kind of identify causalities between those regimes. Okay, basically I have two more particular stories to tell about a few slides. One about biology, one about time series. Shall we continue or?
Yeah, nobody got
sure.
But
Now we are interested. Yeah.
Okay. So, but first, biology. So again, it's a project by my collaborators, but I'm not involved in this particular one. And the question biologists are trying to. Okay, thank you. See you.
Yeah.
Are trying to answer is the following: that in a genome, we'll know that there are genes of different levels of importance, because like in our genome, I think about 80% is just some junk which we picked up during the evolution and which plays no functional role. Some genes are kind of moderately important and they define our fitness level, our competitiveness. And some genes, core genes, are very essential, meaning that a single mutation in this gene will just kill you. And what biologists noticed is that in most of the genomes of and real life beings, essential genes, those which are vital, are clustered in big groups. And they were trying to develop some computational model of evolution, which would show emergence of this clustering of essential genes. But then, okay, the computational biology is computational biology. You deal with some numerical model, and you want to relate this model somehow to the real-life genomes, for which you cannot do simulations, but you want to obtain in your simulation something very similar to what you have in real life, in real biology. And for that, you need to find a metric, a measure, to compare your real-life genomes to the simulated ones, where real-life genome is a sequence of genes, of amino acids, and so forth, and numerical genome is just a sequence of zeros and ones. So how you do it? And again, they played around with just comparing. So since your gene should be viewed as a 1D sequence, because it's very long, it's plotted in a few lines, but you really need to read it from the left to the right. And then close it in a loop. And this one is a markup of a real biological gene from real life, where white pixels designate essential genes and black non-essential. And you see that it's, okay, it's quite random, but indeed there are large clusters. So how do we compare to 1D sequences? One from real life, one from simulations. Just simply computing overlap doesn't make any sense, because individual positions of those clusters do not matter much. It's more important to know what distribution of clusters you have. And first, they try to compare autocorrelation functions of those 1D sequences in one realm and in another one. But it didn't really work out. And then they tried to do our structural complexity thing. But in this case, they did it with moving average. So that I'm not sure exactly what kind of moving window they had. I think about 3 pixels. So that at each step of core graining, they substitute value of this pixel with smoothing average within a window of 3. And in this way, you retain the length of your genome, and you can compare overlaps within the single genome, but at different scales. And then you get this multi-scale structural complexity as a kind of profile. But now with this moving window approach, you can have not just 6 or 7 scales, but you can have a lot of steps, and you get such a curve. So basically, that's CK for genome. And then they defined L1 distance on this CK distributions of partial complexity across scales for real-life genes and for simulated real-life genomes and simulated genomes.
Sorry, for your plot, then very short scales have really increased complexity, but there is a second peak.
Yes.
Frequency in a very long scale. I see. Okay.
Okay, and it turned out, as they told me, a rather good measure, and now it's a work in progress. They're using it to relate their computational models to real-life data. Just to understand, due to, like, for which reasons this clustering of essential genes is important in evolution.
One question, whether it came natural to me, I'm not in genetics. When get the full gene,
yes,
some intervals are really coding like a protein that you want to express, and others are just random intervals. Did you make the complexity only in these subsets, like you could by hand select the intervals of genome that are to express things? And
I need to ask them, because as I told, I'm not a part of this particular project. I'm just telling you about this complexity applications, but yeah, it sounds like a meaningful thing to do. And here I just show a typical simulation within their computational models. When you start with some random distribution of essential and non-essential genes, I think essential are white and non-essential are purple. So each genome, each sequence is a vertical line here, and here is the time. It's iteration of evolution of pool of genomes. And we see that indeed out of some random distribution, those clusters emerge.
Sorry, why does it do that emerge? I mean, what are the rules for this evolution?
Okay, in a very simple terms, like to the extent I understand it, the reason is that mutations tend to occur not on individual sites randomly, but on small subsets of genome. And as a result, if you cluster your essential genes into big groups so that they are neighbors to each other, then the probability for mutation to hit this group and make its genome like reduce its fitness is lower. And as a result, so there is also process of translocation when individual genes can redistribute within genome. And
were mutations the only source of errors, or is also recombination? Because my initial feeling would be like, well, these genes are clustered because you need you don't want to disrupt their occurrence to their core.
I think recombination was also included, but again, since I'm not involved, I cannot answer for sure.
And definition of non-essential, like subjective, or is there an actual definition?
Okay, this definition, it's an actual definition. It's not numerical. Okay, it is computational biology, but in principle, biology is not very numerical. Basically, you can understand it's a gene which mutation, essential gene is a gene which mutation reduces the fitness of representative of your group. Directly reduce.
But recently, the whole idea about junk DNA is changing because we start to find out that the junk DNA is just our lack of understanding what's doing. So
okay.
Yeah, usually in evolution, graphing is for nothing. So there's
No, but still, it's a very interesting thing to look at. But maybe junk DNA is not junk, but core genes are really core. That's we understand very well that if we destroy our core genes, then we're dead.
Sure, there are some basic things
Yeah.
in life.
Yeah.
But then come the genome, that's right.
Yeah, that's related to the previous question, like some chunks will be called specific protein, but the intervals, we don't know anything. But
yeah, so there are theories that indeed the non-coding part, we call it a non-coding DNA, is there to regulate other things like expression and how accessible parts are of the chromosomes. But okay, that's a different story. But it could be something. Makes it more complex.
Okay, and the final story of today, again, is not by me or my collaborators. It is a very recent paper by some people who picked up our notion of multi-scale complexity, and they used it to detect causality relations in bivariate time series. So, okay, personally, I have a mixed feeling about this paper because on the one hand, I'm happy that they use our methods and they claim that it can be used for time series analysis. On the other hand, I would take this part with a huge grain of salt because they claim that they are able to detect causality relations between different time series, and I'm not sure whether they can really distinguish causality from correlations. But let me tell you what I learned from it. So they consider two types of bivariate time series. First, defined by formal dynamical systems, like for example, this logistic model, where we have two variables x and y. They change in time. They affect each other. And for example, here, A is the trends with which y affects x, and B is how x affects y. So, for example, if you switch off A, it would mean that x has some effect on y, but y doesn't really backreact on x and so forth. And then they generated long time series with n elements and truncated them. So they considered just the first part of xy from 1 to n minus tau, and the last part of y from 1 plus tau to n. So the time series, part here, part here. And they asked, can we use complexity to detect causal relations between x and y? And for that, they did some kind of linear algebra transformation. So they converted time series, which are one-dimensional object, into some matrix, P naught, and then computed structure complexity of P naught as a function of time delay tau. And here we have two plots for this logistic map model. In the first case, so please do not look at the solid lines because those are just reshuffled sequences with no correlations at all. And the essential part is those are marked with circles and squares. So in the first case, they picked non-zero A and zero B, meaning that let's see, non-zero A means that y affects x, but x does not affect y, at least explicitly. And then they compute complexity as a function of this time delay. And they discovered that it manifests some non-trivial behavior as a function of tau for negative tau within some quite narrow range, meaning that in the opposite case, when b is non-zero and a is 0, so the effect is opposite now from x to y. This non-trivial dynamics occurs for a narrow window of positive tau, which they interpret as causality relation, because indeed, if you compute this matrix always in the same way, so that X place its own row, Y place its own row, then it's natural to expect causality when from, okay, let's see. Now I stop understanding something, but so if A is non-zero, then Y has some effect on X, and here it can be only if tau is indeed negative, because otherwise Y is in the future with respect to X. And indeed, we have here for negative tau, and when we switch roles of X and Y, we get it for positive tau. This part of the paper seemed reasonably convincing when they got from formal models to real life models of some, for which they used data set of gait of healthy people and people with Alzheimer's disease, so that they put some sensors on their feet, recorded time series representing the gait, and try to understand some causal relations between left and right foot. In this part, I do not believe, because I don't think that they managed to really distinguish causality and correlations. But still, here, the fact that you have only negative tau and rather small tau dependence on the left plot and only positive tau on the right, kind of tells that probably indeed C can be sensitive to some causality.
But is this tau related to the embedding dimension of the reflected?
Sorry, which embedding? No, initially we have just two time series.
I know, but the time series have an attractor.
Yeah.
And Target's theorem tells us that there is some kind of certain embedding direction, the dimension of that attractor. And I'm just wondering whether the value of tau, because what you do, that delayed type of series, whether the embedding dimension is actually related to the tau.
Okay, that's something I do not know, and I think they do not discuss it. So that's why I said that this paper, okay, it might contain some reasonable results, but also I'm a bit skeptical about it. But still, for me, it looks like a kind of invitation to just to take a look at possible applications of complexity for time series. But it's a very good question.
So what you're, but the first sentence there is you combine these, you convert these two time series into a matrix.
Into matrix, and I think it is not just a direct product; it's something more complicated. They have an explicit expression for it.
Okay. Because so what does the last value of X, what is it, N, right? N minus 12?
Yeah.
What does that have to do with the value of Y at 1 plus 12? That's kind of like, the way you write it there seems as if you combine these two into connect them in some one way or another in the matrix, which I don't really understand why.
Okay, it's something we need to understand deeper.
Yeah.
Okay, and basically, it was the last story of today. No, maybe let me just. If you have any more questions, we can continue.
More questions.